Assalamu alaikum warahmatullahi wabarakatuh. Dear students, today inshallah we are going to discuss the data show slides of the digestive tube, which is part of the module 206. This module contains many slides, microscopic and data show slides. First, we're going to discuss the oral cavity. We're going to take from the oral cavity the lip and the tongue. Regarding the lip, as you can see here, this slide, I'm having a section. Surface contains skin, which is covered with keratinized stratified squamous epithelium, and surface contain mucous membrane, non-keratinized stratified squamous epithelium. Between them there is muscle, skeletal muscle, and near the mucous membrane there are mucous glands. Okay, for the first look here, I think you can misdiagnose this section as the eyelid, because both of them, one side skin, one side mucous membrane, between them skeletal muscle, and near the mucous membrane there is glands. How to differentiate? Look at the margin or the border that present between the skin and the mucous membrane. Here in the lip, it's called the red margin. It does not contain any hair follicle. While the eyelid, there is eyelashes. Here you don't have any hair follicles. So a section of the lip, here the skin, the hair follicle, sebaceous glands, the muscle, which is skeletal muscle, orbicularis oris, and the glands, labial glands, and here is the mucous membrane. Here, dear students, you can see here the region of the lip. They contain the skin, keratinized stratified squamous epithelium. This is the hair follicle, and the sebaceous gland related to the hair follicle. Again, here this is the skin, and here is the muscle. In the inner surface of the lip, I can see the mucous membrane, non-keratinized, stratified, squamous epithelium lying on clear wavy basement membrane, and here is the connective tissue or lamina propria, and here is the labial glands. And there are blood vessels here. This is the labial glands. There is a section in the muscle which is known as orbicularis oris muscle, and this is the mucous membrane. As you see here, dear students, this is the red margin. Between the skin and the mucous membrane, there is a margin known as the red margin, covered with keratinized stratified squamous epithelium, does not contain any hair follicle. The second slide is the tongue. One side covered with keratinized stratified squamous epithelium in raised papillae. This is the papillae, which is raised epithelium and underlying lamina propria or connective tissue present at the dorsum of the tongue. Here is the muscle directed in different direction, and the mucous membrane, non-keratinized stratified squamous epithelium, or the ventral surface. Again, there are glands near the ventral surface. Here, dear student, this tongue contains mainly filiform papillae, contain many filiform papillae and some fungiform papillae, and this is important as this is tongue human. This is tongue human. This is the filiform papillae covered with keratinized stratified squamous epithelium, and this is the raised papillae. Appears conical in shape, and here is the muscles and the lingual glands. Here another type of papillae known as fungiform papillae. Okay. The third type of papillae is known as circumvallate papillae. You can see here that's not raised above the surface and surrounded with a valley and contain many taste buds. They are present at the sulcus terminal between the anterior 2/3 and the posterior 1/3 of the lower stem of the tongue. Circumvallate papillae. And here are the ducts. The glands, which is known as the von Ebner, is purely serous, open at the duct, and the duct open at the valley in order to clean or to wash the taste buds from the substance that I'm going to taste, if it's salty, bitter, sweet, and so on. Circumvallate papillae. This type of papillae not present in human, that it's called papilla foliata. Papilla foliata, it's present at the tongue of rabbit, and this is not present in human. We can say the rule: once papilla foliata, always tongue rabbit, not present in human. As you can see here, it contains many taste buds, and this is the muscles. Number 4 is the striated muscle. Number 3 is the serous gland. Number 2 are taste buds, and number 5 the ducts of the salivary glands. You can see here the taste buds. We took it from the papilla foliata. You can see here the taste buds. It's formed of three types of cells: the supporting cell, the taste cell, and the basal cell, which act as a stem cell. So here, the oral cavity, under light microscope, you can see only the lip and the tongue human. While data show slides, you're going to see the tongue human, which is formed mainly of filiform and fungiform. You can see the circumvallate papillae and the fourth papillae, which is papilla foliata, which is not present in human. And in the data show slides, you're going to see also the taste buds. Under the microscope, you're going to see only the lip and the tongue human. Regarding the esophagus is a long tube. Okay, it is a long tube. I'm going to cut it TS section so it will appear rounded structure with lumen. And please focus on such section, because you have many section rounded structure with lumen, and I'm going to tell you how to differentiate under the microscope. This is esophagus. This esophagus is for human or esophagus dog. From where I know, I'm going to tell you. As I told you, you're going to have a rounded structure or oval structure with lumen. How I'm going to differentiate? Look please at the lining epithelium. The lining epithelium is non-keratinized stratified squamous epithelium. Okay. How do I know it? It is clear, it has clear wavy basement membrane, and I'm having nuclei of different shape. First columnar oval nuclei, then many rounded, then flat nuclei. Okay, look here, dear students. Under the epithelium there is lamina propria. Okay, and there is muscularis mucosa. What is muscularis mucosa? It is muscle layer, smooth muscle layer, inner circular and outer longitudinal. It leads to movement of the mucosa. Then in the submucosa, I'm having gland. What type of gland? Mucus gland. I'm having mucus gland in the submucosa. Then I'm having skeletal muscle, inner circular, outer longitudinal. Then the outer cover is the adventitia. Okay, so if I see between the epithelium and the muscle layer glands, it is esophagus dog. Again, it's formed of mucosa, submucosa, musculosa, and adventitia. What you can see here is esophagus cat. Round structure with human. Okay. After the epithelium, lamina propria and very well developed muscularis mucosa. Between the epithelium and the muscle in the submucosa, I don't have glands, so it is esophagus cat. Again, I'm formed of 4 layers: mucosa, submucosa, musculosa, then adventitia. Look, between the epithelium and the musculosa in the submucosa, I don't have glands, but I'm having very well developed muscularis mucosa, esophagus cat. Here is section in the epithelium, which is non-keratinized stratified squamous epithelium, and this is the lamina propria. So in the esophagus, data show I'm having esophagus dog and esophagus cat, while the microscope I'm having only esophagus dog. In the stomach, I'm having a section like this. Okay, so I'm having strip. Under the microscope, I'm having 2 slides, fundus and pylorus. While in the data show slide, I'm having another 2 slides, which are junction between the esophagus and the fundus, gastroesophageal junction, and between the fundus and between the pylorus and the duodenum, pyloro-duodenum junction. But under the microscope, I'm having only 2 slides. The fundus, first, the fundus. It's formed of mucosa, submucosa, muscle layer, and the serosa. Okay, what is this? The fundus is highly folded. Okay, and what the crowd this? It is the fungal gland. I'm going to discuss it in detail now. This is the submucosa and the muscle layer, smooth muscle, but there is an extra layer, inner oblique, middle circular, and outer longitudinal. Okay, I want to study the gland. The fundal gland here, they are crowded. This is the pit. Pit to gland ratio is 1:3 or 1:4. Dear students, look here. This is the pit. Pit to gland ratio is 1:3 or 1:4. The pit is narrow and short. I can see here different cells. I'm having surface epithelial cells, and I'm having the mucus secreting cells, mucus neck cell, the parietal and the peptic cell. So I'm having different types of color. The mucus secreting cell appears pale, the peptic cell appears dark basophilic, and the parietal cell appears dark acidophilic. So number 1 is the surface mucus cells. Number 2 is the pit, and here is the glands. In the gland, what I can see, dark basophilic cell, which is the peptic cell, dark acidophilic, which is the parietal cell, and slightly stained cell, which is the mucus neck cell. Again, dear students, this is the pit, characterized by being narrow and shallow. Okay, and these are the fungal glands, which are crowded. Less connective tissue corium can be seen between the cells. Again, dear students, this is the fungal gland. Shallow, narrow pit, and this is the glands which are crowded, and I can see dark acidophilic and dark basophilic cells. The dark basophilic cells, which is the peptic cell, I can see it in the lower part of the gland. And this is the muscularis mucosa, and this is the submucosa. Yeah, dear students, is a very nice photo. It is light microscopic picture. I can see the dark basophilic cell, which is the sheath cell or the peptic cell, and dark acidophilic cell, which is the parietal or the exanthic cell. Here, number one is the parietal cell, and number two is the peptic cell, and this is the muscularis mucosa. Again, dear students, this is fundus gland. From where I know, the pit is narrow and shallow, and I can see the fundus glands. This is the muscularis mucosa, submucosa, and the muscle layer. This photo is not nice. Why? Because I cannot see the crowdness that present at the connective tissue corium. I can see many connective tissue corium, but from where I conclude that it is a fundus because of the narrow shallow pit. Here, dear students, is the pylorus, a very characteristic picture of the pit. Very wide and deep pit. The pit to the gland ratio, 1 to 1. Another very important feature here, dear students. The cells, mainly mucus secreting cells, so appears vacuolated. The cells, mainly mucus secreting, so it appears vacuolated, and I can see connective tissue in between the glands. Again, this is the pit, wide and deep. Pit to the gland ratio 1 to 1. Most of the cells are mucus secreting cells, so the cytoplasm appears vacuolated, and it is not folded as that of the fundus. Again, dear students, this is the pylorus, from where you know wide deep pit, deep pit to the gland ratio 1 to 1. The gland mostly formed of mucus secreting cells, so the cytoplasm appears vacuolated and there is connective tissue between the glands. This is the muscularis mucosa. This photo is very nice because it mimics the light microscopic picture that you're going to see. The mucosa not folded, the pit very wide and deep, the glands mainly mucus secreting cells. This is the muscularis mucosa and the submucosa. And this is a very big muscle layer. It's formed of 2 layers, but the inner circle is very big because it formed the pyloric sphincter. Inner circular, outer longitude. So, for the stomach, under the microscope, I'm having only the fundus and the pylorus, and I want to differentiate between them. How? First, the fundus, the mucosa is folded, the pylorus not. Second, the fundus glands are very crowded, while that of the pylorus less. Number 3, the pit to the gland ratio in the fundus is 1 to 3, and in the pylorus 1 to 1. 4. Most of the cells present in the pyloric glands are pale, as they are mucus secreting, while that of the fundus colorful, darkly acidophilic, darkly basophilic, and pale. The muscle layer of the pylorus, the inner circular layer, is very big, forming the pyloric sphincter. For data show slide, we have the fundus pylorus and gastroesophageal junction. Gastroesophageal junction, long strip. From one side here, I can see as if I'm saying the esophagus dog, non-keratinized stratified squamous epithelium. In the submucosa, I'm having mucous glands, and this is the muscle layer, inner circular, outer longitude. While here, near the stomach part, I'm having change of the epithelium from non-keratinized into simple columnar, and I'm having glands, gastric glands. I'm having pit, having crowded glands. The submucosa does not contain glands as that of the esophagus. In the muscle layer start to appear an additional muscle layer, which is the inner oblique, middle circular, outer longitudinal. Again, dear students, here is the side of the esophagus. Number 1, non-keratinized stratified squamous epithelium. Number 2, it's near the stomach. Number 3 is the esophageal glands. There's no glands in the submucosa near the stomach part. And inner oblique appears. Here is middle circular outer longitudinal. Here inner oblique, middle circular outer longitudinal. I'm going to discuss the small intestine, and I'm going to have duodenum, ileum, and the pylorodduodenal junction. Small intestine, we are going to study the duodenum and the ileum. Duodenum and the ileum and the pylorodduodenal junction. In order to understand the stomach, the small intestine, you know that it's involved in after digestion, absorption. So in order to do absorption, I want to increase the surface area, so there are villi. And in the villi lined by cells containing microvilli. All this increasing the surface area. I want to tell you that the small intestine, look here dear students, the small intestine contains villi and crypts. Villi and crypts. How to differentiate between villi and crypt? The villi contain nothing in between, while the crypt containing connective tissue in between, the lamina propria. Okay, so here a villi. From where you know that this is villi, does not contain anything between. Villi. Lined with simple columnar absorptive cell contained microvilli and goblet cell. And this is a connective tissue corium. This is the connective tissue corium, like a finger. Here is the crypt. From where, doctor, we know that this is the crypt? From where? I can see connective tissue in between the crypt. Lined simple columnar with goblet cell. In the crypt, I'm having a cell known as a Paneth cell, characterized by basal basophilia and apical acidophilia. From the small intestine, I'm going to study the duodenum. Okay, the duodenum is rounded structure with lumen. Look at the lumen. I can see what? I can see villi and crypts. How, doctor, you differentiate between the villi and the crypt? The villi contain nothing in between, while the crypt contain connective tissue. Okay, how could I differentiate the duodenum from the ileum? Between the villi and crypts and the muscle layer, there is submucosa containing glands. These glands known as Brunner's gland. Mucus secreting glands. Again, here is the villi and this is the crypt. From where you know? In between the villi, nothing. In between the crypt, connective tissue. Between villi and crypt and the muscle layer, there is submucosa containing Brunner's glands, which are mucus secreting glands. Here is the ileum, large rounded structure with lumen. Between the lumen, villi and crypts, and the muscle layer at one side, there are collection of lymphatic follicles known as Peyer's patches. Again, here is rounded structure with lumen. I can see villi and crypts. So it is one of two, duodenum or ileum. This is ileum. Why? Between the villi and crypts and the muscle layer, at one side, there are the Peyer's patches, which aggregates of lymphatic follicles. Again, dear students, these are the villi and these are the crypts. Between the villi and crypts and the muscle layer, there are collection of lymphatic follicles known as Peyer's patches. a long strip, so it is junction. A very characteristic thing here, this elevation, this huge hump, which is the pyloric sphincter, which is the inner circular layer. In here, near the pylorus, I'm having pyloric glands with wide, deep pit, and the submucosa contains nothing. Here near the duodenum, I'm having the Brunner gland here, and the muscle is less. The Brunner gland, which is a mucus gland present in the submucosa. So, again, we have the slides of the small intestine. I should differentiate between the ileum and duodenum, and I'm having long strip pyloroduodenal junction, but the pyloroduodenal junction is data show slide only. The last thing we're going to discuss, inshallah, is the large intestine. I'm going to study the colon and the appendix. The large intestine or the colon, I am crypts only. I don't have villi. Crypts only, I don't have villi. Very regular and contains many goblet cells. Mucosa, submucosa, and muscle layer, inner circular, outer longitude. No villi. And the crypts are wide and deep. Higher magnification, here is the crypt. Contain many goblet cell. No villi. Here the crypts contain many goblet cell, no villi. And this is the connective tissue corium in between. This is the muscularis mucosa and the submucosa. How to know the large intestine? Number 1, no villi. Number 2, wide, deep crypt. Number 3, many goblet cells. I can see additional thing? Yes. Tinea coli. The muscle, inner circular, outer longitudinal. The outer longitudinal arranged in 3 bundles known as Tinea coli. 1, 2, 3. And sometimes I can see fat, which is known as appendices epiplewicae. This is the appendix. Only crypts. Round the structure with lumen, only crypts. And the lymphatic follicle all through. This could be differentiated from the ileum. Yes, the ileum contains villi and crypts and the lymphatic follicle at one side. Here I'm having crypts only and the lymphatic follicles all over. This is appendix rabbit. Lymphatic follicle all over and the crypts here appears as Y-shaped crypt. Now, dear students, we finished the data show slides and discussing the microscopic slides of the GIT, gastrointestinal tract, and then we're going to discuss the questions after it in the next video, inshallah.